Introduction. The nerve impulse is triggered by synapse from a presynaptic neuron. Presynaptic neurons can connect to postsynaptic neurons via 1. Dendrites, the fibrous extensions of the neuron which connect it to the presynaptic cell. 2. The soma, which is the body of the neuron, containing the nucleus. 3. The axon, which is a longish fiber joining the cell to the subsequent neurons. In this simplified model, the potential traverses the cell, starting from the dendrites, passing through the soma, progressing to the axonal terminals which connect the cell to the subsequent neurons and pass on the impulse by releasing neurotransmitters. A neuron can have more than 1,000 connections with neighboring cells. EPSP diagram. The diagram shows a frame of some 6 milliseconds. Commencing as ligand or voltage gates initiate a potential firing of the neuron to the return to the resting potential. Note that the potential, the region of depolarization, travels along the axon as a saltatory wave. It is not an instant reaction. A neuron's length can vary from less than a millimeter to more than a beta. In humans, nerve conduction velocity can vary from less than 1 meter per second to some 100 meters per second, depending on the type of neuron. Gates and ions. Pores in the cellular membrane of the neuron, known as gates, are the primary engine to modify the potential of the axon. Positive cations, sodium, potassium and calcium, and negative anions, chlorine, are diffused across the axon's membrane to modify the potential of the axon. Transmission and propagation of the impulse. The phases of neuronal excitation are: resting potential. A neuron at rest has a potential of -70 mV. Neurotransmission. The impulse starts with a chemical signal originating from the presynaptic neuron. This chemical signal causes ligand-gated channels in the postsynaptic neuron to open EPSP/IPSP. The threshold potential of a neuron is -55 mV. If the threshold potential is reached in the axon, the impulse is propagated and voltage-gated channels are activated. Depolarization. Sodium gates open. Positive ions, Na+, flow into the axon via sodium gates and raise the potential. If the threshold of -55 mV is reached, the potential is maintained. Repolarization. The sodium gates close and potassium gates open. Positive ions, K+, flow out of the axon via potassium gates and lower the potential. Hyperpolarization. Repolarization results in a drop of the potential to -100 mV. Return to resting potential. Specific gates, known as pumps, rebalance the axon's millivoltage back to the resting potential.